So welcome back to step 2. So at this point, our virus is in the host cell, and the question is, what does it do once it's inside the host cell? Okay. So first of all, viruses can only reproduce in the host cell. They themselves cannot reproduce. So they cannot reproduce themselves actually outside of the host cell, like they're nothing. They can only cause damage and be infectious and all that stuff when they're inside the host cell. Okay. For that reason, because they need the host cell to reproduce, they're actually not considered living things. So viruses are not considered living things. Now, once in the host cell and their DNA is in the host cell, they use the enzymes, ribosomes, and other molecules like tRNA to make more of themselves. Now the question is how. So this is studied this was studied mostly in bacteria cells. And bacteria phages, I can never say that word correctly. Tommy put that on the list of things I can't say. This is another word for viruses. So this word here is another word for viruses, and these are viruses that infect bacteria. And they're the most studied of all viruses, at least in the beginning of learning how viruses infect cells. And they go through, once inside the cell, they go through 2 possible ways, okay, that they can infect the cell or reproduce themselves. The lytic cycle and the lysogenic cycle. And we're about to go through those 2 things. So, in the lytic cycle, okay, the lytic cycle ends in the death of the host cell. And in the lytic cycle, okay, the new phages or new viruses are produced, okay, and they eventually explode the bacteria and release the baby viruses. And this baby in quotation marks, like viruses don't grow up, okay? And then these baby viruses, okay, that are released, then go on to infect other cells. Okay, now we're going to look at this in a picture. Alright, so if we see here, in step 1, we have this virus, okay, and it's injecting, in this case, it's latched on using its glycoproteins to the bacteria cell, and it's injecting its DNA into the virus. So here, it attaches to the cell, and here it injects its DNA into the bacteria cell. So this is a virus injecting its DNA into the bacteria cell. Then, okay, is the ribosome, the tRNA, the RNA RNA polymerase. Those things are used to take the code in the viral DNA and turn it into the proteins in that viral DNA. So here we can see like the parts of the virus are being assembled. Okay, and then last but not least, in the bacteria cell, the parts that it just made of the virus. It replicated the DNA of the virus. It turned all the genes in the virus into protein. It assembles those viruses into new virus particles. And then when there's enough of those virus particles, they explode the cell. Okay? Then, those viruses can now go on and infect other bacteria cells. Okay? Now think about this. When you have like the flu or the cold, this is what's happening. Is that the virus gets into your body somehow, either from having the virus on your hand and going to your nose or going to your mouth, or it could get, if it's a sexually transmitted virus, it could get in through sexual contact. And then once that virus gets into your cell, it infects cells, like here, then uses your cells to make more of it, then it explodes your cells, and then these viruses then go on to infect other cells, and make more of themselves inside your cells and explode your cells, and then go on and the cycle continues. When you finally feel sick is when enough of your cells are exploded, and enough of your cells are spending their entire life making viruses instead of doing what they should do. That's when you feel sick. So now, we went through the lytic cycle, which is where, I'll summarize one more time. The virus gets its DNA inside, okay? Then it uses the ribosomes, the tRNA, the RNA polymerase, the DNA polymerase to replicate itself. And remember, those things are from the host cell. Then it assembles the virus particles and explodes the cell and goes on to infect other cells. Now, we are now going to talk about the lysogenic cycle, okay? Now, in the lysogenic cycle, instead of exploding the cell, the DNA of the virus, the phage genome, gets incorporated into the host's DNA and stays in there and lays dormant. Dormant means like it sleeps in there. Okay? And then when environmental conditions are right, it launches into the lytic cycle, and then does that whole thing we just discussed. So now we're going to look at the lysogenic cycle. So we're starting here in this cycle, and at this point, this like red circle is the virus or the phage DNA that's already been injected into the cell. And we're doing the lysogenic cycle right now. So instead of this DNA being used immediately to make virus particles, this DNA incorporates itself into the genome or into the DNA of the cell it's infecting. And since that DNA is in there, when the cell, in this case is a bacteria cell, replicates, it replicates the viral DNA. And then you have many, many, many bacteria cells with this viral DNA in it. Okay, then we're going to skip step 5. We're not going to talk about here. So now we have all these cells, these bacteria cells that have been infected with a virus, and inside their DNA, they have virus DNA in it. Now, what can happen, and it's not really well understood what causes it to happen, okay, is that some sort of environmental condition, heat, cold, can cause these cells that have the viral DNA in it to enter the lytic cycle. Okay, so this cell can come, and I may need my pointer again. This cell can, now it has the viral DNA. These are actually 2 cells. The viral DNA is here, and the viral DNA is here. These cells, when environmental conditions are right, can enter the lytic cycle. Okay, here, I guess here. We're step 1, so I'm lame. Here. So again, but the DNA is already in here. So in this case, this cell has the viral DNA here. And then all of a sudden, when environmental conditions are right, okay, it can all of a sudden take that viral DNA, okay, and start making baby viruses. Just like it did in the lytic cycle. Okay, but the point to know is that in the lysogenic cycle, if the virus is doing a lysogenic cycle, it doesn't make more baby viruses. It just inserts its viral DNA into the bacterium cell, and it stays there until it's ready to enter the lytic cycle. An example of this that we've seen in humans, and it's funny, you guys are too young or to like remember this or be affected by this, but you have all at this point gotten a vaccine for the chickenpox. Okay? When I was a kid, there was no vaccine for the chickenpox. So I got the chickenpox. And one of the things that the chickenpox does is it injects some of its viral DNA into my cells. And this happens if you've gotten the chickenpox, when you get older, there's some viral DNA left over from the chickenpox in some of your cells. You can get something called shingles. And shingles is when, for whatever reason, it infects your nerve cells. Your nerve cells feel like they're on fire. And you feel pain even when nothing's touching you. So because I've had the chickenpox, and there's probably some of that viral DNA resting in my cells, usually happens in your 60s and 70s, I'm at risk for getting shingles. Because I have viral DNA inside that's left over from my chickenpox infection, which you guys have all had the vaccine for. So, you're safe. Okay, now, the last thing we have to talk about in this lesson is viruses that have RNA as their genetic material. Okay, the viruses that we've been talking about so far have DNA as their genetic material. Okay, many animal viruses, so viruses that infect us, have RNA as their genetic material, and they're called retroviruses. Okay, one of the famous viruses that is a retrovirus that has RNA as its genetic material is HIV. Now, all the steps that we talked about, they have one more additional step that they need to do to infect cells. Okay. They have an enzyme inside themselves called reverse transcriptase. And because their their genetic material is RNA, they need to copy their RNA into DNA before they can integrate their DNA into us. So they have an additional step. They have to use the enzyme reverse transcriptase, okay? And that copies the RNA into DNA, so that the DNA can then go inside our nucleus. So this is the reproductive cycle of the HIV virus, which again is a retrovirus because it has RNA, and I can't write, as the genetic material. So here, in the cycle, the first thing that happens is that like the HIV virus attaches to, and the HIV virus infects immune cells. So it white blood cells specifically. specifically helper T cells, if that means anything to you. We're going to get into that, so don't worry. And it injects its DNA, RNA, whoa, it injects its RNA, the RNA is the red, into the nucleus, and it also injects these, like, little, like, purple balls. Those are some capsid proteins that it has, but that's not the point. The point is, the first step is that the HIV virus gets its DNA, its RNA, gets its RNA into the cell, and it infects immune cells. Now you'll see we have this viral RNA here, and it needs to be turned into DNA. And that's done by reverse transcriptase. Then, and it actually makes double-stranded DNA, so it copies it twice, okay? Then, the DNA gets inside the nucleus and incorporates itself into your DNA, okay? Then, that strand, the viral DNA that's incorporated itself into your DNA, gets transcribed into mRNA. And then, as we learned, I just had a quiz on, that mRNA gets turned into protein, okay? And then the virus particles are assembled, and then the virus particles exit the cell and go on to infect other immune cells. Now, people who have HIV do not die of HIV, okay? They die of a disease called AIDS, okay? Now, why people with HIV die, okay, is because these viruses are infecting their immune cells. Eventually, they have very, very few immune cells. They call it the white blood cell count has gone down. And then they're at risk for getting all sorts of other illnesses because their immune system is so suppressed that they can't fight it. So HIV doesn't actually, the virus itself doesn't kill people. People with HIV, which HIV die because their immune system can't fight off normal illnesses that your body fights off every day. And instead, they die of them because your immune system is so suppressed it doesn't win. Okay. The last thing we have to talk about is the evolution of viruses, or why it's hard to both treat and find vaccines for viruses. So, viruses have fast mutation rates because when their DNA is replicated, there's no checking mechanisms to check DNA polymerase. And if you remember, DNA polymerase makes mistakes. So in your cells, when it's your DNA, it's checked. But viral DNA is not checked. So those mistakes, instead of getting fixed, get passed on. Now, RNA viruses, okay, have even faster mutation rates. Okay? That's because reverse transcriptase is even more error-prone than DNA polymerase. So there are more mutations that are passed on and not fixed. That's why it's extremely hard to get a vaccine or drug that works to treat all strains of the HIV virus. Because they have this enzyme that makes a lot of mistakes, they have a lot of mutations, which allows them to adapt, okay, and become resistant to the drugs that we use to treat the infection. Okay? It also makes the development of vaccines difficult because these viruses are always getting new mutations, okay, that evolve them to evolve and escape our vaccines or drugs. Okay? Because the viruses that do not escape our vaccines and drugs die, okay, but those that get a mutation that helps them escape the vaccines or drugs live. And then those HIV viruses, or those strains of the HIV virus, are resistant to the viruses, to the drugs and vaccines that we have. Tomorrow's Do Now is going to focus on some of the more difficult parts of viral infection. But do the guided questions, and I'll see you tomorrow.